Teresa, and thank you, Jerry, for the kind invitation to come. I feel kind of, a bit overwhelmed and embarrassed by the kind things that Bob Nussbaum said, and I hope that I can make the research efforts that are going on kind of accessible and that you find this a useful session. So I'm talking about my research as well as that of other people to try and give you an overview. Because I think there's a real gap in communication between researchers and families, and is such technical information. I think Dr. Brewer did a fantastic job yesterday of trying to make that more accessible. And as I transitioned from really being a basic biochemist and cell biology to having this new arm of research interacting with all of you, I wanted to try and bridge that gap in some way. And so we started this video project, to Teresa's wonderful logo here, that's centered on the patient, that brings researchers, organizations, and families together. And hopefully this, for all of you, this video resource is something that you can share and use with other people to help communicate and tell stories. And also useful for me, for when new researchers join my lab, to actually give them the reason why we're doing this work, and that really this is an urgent problem, and how their work can fit into benefiting society as a whole. Because I think as scientists and cell biologists, it is very easy to lose sight of that underlying basis of why we're doing what we're doing. So this project gave a training in filmmaking using mobile phones, and thank you for the participants here, the Otani family there, who were involved in this, and it really was a privilege working with you all. So this has been an opportunity for storytelling. There were professional editors that worked on these videos, and Teresa and I and our public engagement team at the Gurdon Institute recently wrote a paper to disseminate this work to the research community. So we've currently got 4 videos. I'm going to show them spread out through the talk to kind of break up the science a bit for you.
We are the parents of boys with Lowe syndrome. Lowe syndrome is a 1.5 million diagnosis, a rare genetic condition that causes a number of medical complications, physical and cognitive disabilities, and developmental delays. Each diagnosis is unique and complex in its own way.
Thomas, can you see stuff? What are you looking at?
Having a child with Lowe syndrome is complicated. They are your whole world, and they depend on us to be their biggest advocates.
We become our child's specialists, spending hours at doctor's appointments, therapies, managing medications, and arguing with insurance companies. It is constantly working to balance the medical needs with taking time to enjoy watching them grow and learn.
Look at you! There are good days and bad days. With Lowe syndrome, you never know what the next day will bring. We savor the small victories that we were told might never happen. Not all with Lowe syndrome are impacted in the same way. Some are more independent and verbal, while others may have several physical and cognitive delays.
Some endure more serious medical complications that can be life-threatening, while others may not need as much intervention.
In younger years, parents are exceptionally busy with education, medical treatments, and therapies. Some are fortunate enough to have a family network of support. However, no matter what your support system, you are your child's advocate since they are unable to advocate for themselves. As they become adults, coordinating medical appointments and daily programs can become more routine.
However, daily assistance and monitoring is still a big part to ensure their safety and well-being because there may be limited understanding of the world around them.
Our hopes for the future are that our children will grow and thrive despite living with Lowe syndrome. We want our families to have strong support systems and access to effective medical services. We need healthcare and school systems to be more compassionate and less traumatizing as they care for our children.
We hope our boys will walk one day and gain independence to explore the world on their own terms and be known for more than just their diagnosis.
As parents, we want others to understand that we must take care of ourselves so we can worry less about who will continue to care and advocate for our boys when we no longer can. Our dream above all is to find a cure for Lowe syndrome.
We do not know if a cure will be discovered in our sons' lifetimes,
but we hope and wish the best for all Lowe syndrome families and those that will come behind us. All done?
So I had a great response when we released these videos. People were coming up to me in the Institute and saying, oh my goodness, what can we do to help? And I didn't realize, it was so bad. And what can we do to help? And I'm using it to help my researchers understand and keep working, on trying to find something to help. So our genes are the set of instructions or blueprints that are found in all our cells. So whether it's our brain or whether it's our kidney, these organs are made up of cells, and the DNA, the genetic material, is in the nucleus. So here I'm showing a neuron here, it's in your brain, an astrocyte cell, and it might be something to do with the difference with different pathways to get to astrocytes or neurons that Raghu Pandit is working on in India that might be an underlying basis in the brain. We understand most about what's happening in the cells in the kidney. So from Eileen Brewer talked yesterday about the proximal tubule. Here I'm showing 3 cells representing the proximal tubule within the kidney. This is an epithelial layer where these cells have to take up substances from the urine, that reabsorption process. And so these cells all have the nucleus in them where the DNA, the genetic material, lives. And so when there's a mutation in that DNA, that mutation is present in all the cells of our body. And depending on what the cells need to do depends on whether that gene mutation is affecting that particular tissue. So my talk today is going to be in 3 parts. And I've got a kind of color coding so you can navigate where we are, particularly if you want to go back and watch the video later. So we've got the red section here, the turquoise section, and the navy blue section. And the first section is how the OCRL gene mutations give rise to the symptoms. And here I've got an opportunity to show the second video in the series.
Lowe syndrome is a rare genetic condition affecting approximately 1 in 500,000 people, most of whom are male. A genetic condition is caused by an alteration or mutation in the DNA sequence. In Lowe syndrome, the alteration occurs in the gene OCRL1 and can impact several organs, the eyes, the brain, and the kidneys in particular. While those living with Lowe syndrome are not all the same, most are affectionate and sociable, love music, and have a great sense of humour. The condition can lead to physical and cognitive disabilities and various medical issues. Nearly all individuals with Lowe syndrome are born with cataracts, usually removed within the baby's first few months. About 50% may develop glaucoma as they get older. Symptoms range from mild to severe and can include intellectual disability, short stature, poor muscle tone, delayed motor development, seizures, behavioural issues, as well as bone problems, joint difficulties, and dental issues such as gum problems. Kidney problems typically emerge from around age 1 and tend to develop over time. Reabsorption in the kidney of essential substances such as salts, amino acids, and proteins is poor, causing complex nutrient deficiencies. While there's no cure, symptoms can be managed with medication, surgery, occupational therapy, and adapted education. Researchers continue investigating the role of the OCRL1 gene and the mechanisms of Lowe syndrome, and are working on developing new therapies. Reach out and support the Lowe syndrome community.
So the OCRL1 gene, there's the genetic mutation, causes the production of an OCRL enzyme. So all of us produce enzymes that are made up of amino acid sequences that are encoded by our genes. And this is where the mutation comes, and so then there's a mistake that results in the error in the OCRL enzyme. So that OCRL enzyme converts one fatty acid substance into another. So it's present at cell membranes. So these are the barriers between the inside and outside of the cell. This could be at the cell membrane, at the wider outside area, or also at intracellular organelles inside the cells. And so it acts a bit like a traffic light system. This is called phosphoinositide signaling, where these what are called head groups here of these fatty substances that make up our membranes are turned from one type of chemical identity into another type of chemical. And so if you have a mutation in the gene, so it's like a spelling mistake in the gene, you can see we normally read it out here, it makes sense, but then it doesn't make sense when there's this mutation. That leads to a change in the A, T, C's, and G's. So if you have your genetic mutation information, that will be in that form of the letter and a number that tells the scientist whereabouts, what it is in the gene that's changing. So that genetic mutation, it's transcribed and then it's translated to give the code for the amino acid sequences. So this is this ASP-CER-GLY-LU sequence here, and there's many, many of them in large strings. So those large strings, they then fold up to form sort of molecular machines that have a particular shape. And that allows them to do this chemical conversion step of one fatty substance into another one. So you can think of it as a bit like origami, the pictures that I've got here, that the DNA sequence makes the amino acid sequence that then folds up to make that protein, that molecular machine that can do a particular job. So these mutations can take on different types. So it could be that like the wings are cut off or the tail's cut off. It could be a truncation mutation that we call that. It could be a misfolding mutation, so that's indicated by this picture here. And often when proteins are misfolded, they're then gotten rid of by the cell. And so it ends up being a total deficiency or a null if you have a folding mutation. It can also be the kind of mutation that changes the particular chemistry of how that lipid, that fatty substance is converted from one to another. That's called an active site mutation, where it can't do the chemical change that's required to make that transition. And so this can come, for example, instead of having a serine here at one of the positions, you might have an asparagine instead, this kind of different chemical that has a different chemistry and can no longer do that modification. So if the OCRL enzyme is dysfunctional, so that enzyme comes from the gene that has the mutation, it can mean that these special fats are no longer converted. And we know that one particular site of where those fats are converted is in the proximal tubule cells of the kidney. So this is the cells that are best understood. So this is the urine that's forming here within the kidney. It's the blood's being filtered through the glomerulus. And then, as Eileen Brewer talked about yesterday, the substances need to be taken up by those cells. One of the things that we look at as a marker for that uptake is the protein. And what I'm showing you here in these green shapes are what's called receptors. These go and grab the protein out of the urine, and then it's taken into the cell. And the way in which that's taken into the cell is that bending, that deformation of the membrane to form a sort of bag, we call it a vesicle, platinum-coated vesicle, that has those receptors in it with the protein that needs to be taken up. So the proximal tubules are an amazing machine at doing this endocytic and endosomal trafficking pathway. And through that pathway, this lipid identity, this fat identity, needs to be changed through that pathway. So that you can go from this pink membrane here to this turquoise one here. And then what happens is those membrane trafficking compartments, these are recycled back up so that you get the receptor back onto that membrane surface to go and grab more protein and keep doing it. So what happens when you fail to get that change in the fatty substance through the membrane trafficking pathway is you get, for us they look big, big with looking down the microscope, aggregations of a protein called actin around those endosomal intermediates, and that's indicated here with this sort of orange crosshatch pattern. And that's what I've been working on for the last 20 years, is how these specialised fats are converted from one to another, and what are the consequences of that, and how is that leading to aggregation and assembly of these actin filaments. So you can think of this as being like a traffic jam in the uptake pathways within the kidney. That's the best understood. There are other things that this is doing as well, that there are other complex effects on cells that we're still trying to work out. In particular, it's unknown which of these cell biological changes are most important in each organ. So there's a lot of membrane trafficking that goes on in the brain, but it's not known whether it's a membrane trafficking basis to what's going on in the brain, or it could be some of these other effects that we're also seeing down the microscope. Okay, so that's the finishing of part one of the talk, and now I'm going to move on to part two, which is the candidate treatments that are being explored. And the reason why I took you through the things that I did in the first section is because these different treatments that are suggested affect these different levels of organisation that I described. That there's the gene, there's the enzyme, and then there's the cellular consequences of the mistake that's arising from that enzyme dysfunction. So the candidate treatments are targeting these different levels. So there's gene therapy, which is aiming to add on a gene or fix the gene. There's targeting the OCRL enzyme to making it do that chemical conversion better to prevent this misfolding process. And then there's drug repurposing efforts, which are looking at these cell biological changes directly and finding other ways of changing the way the cell's functioning to correct that blocked uptake pathway. And so at the moment, these are focused on the kidney. So because it's caused by a genetic mutation, gene therapy is conceptually very appealing because it seems as though it gets to the real heart of the matter, that there's this gene mutation. So it aims to replace a defective gene with a normal one by adding it into the cell. CRISPR technology is aiming to repair the defective gene, or you can also change another gene to help the original one. So we know in the case of OCRL that there are many different mutations that give rise to Lowe syndrome. And so if we're going to think of individually repairing each of those different mutations, that's going to be a massive job. So what's happened in some diseases is that you instead change another gene that helps compensate for that loss. So it requires different treatments for each organ, and that's because of the inherent constraints of trying to get a big gene into a cell. So we've got all these genes are in all our cells of our body, and that's not going to be possible to change all of them. So it's targeted to specific organ systems, and so that means it's more straightforward if you've got a disease that's only affecting one organ system, whereas in Lowe syndrome you've got these three main sites. So it was very exciting in mid-May, the reports of the in vivo gene editing that came out of CHOP. And this is using what's called a platform approach of CRISPR and lipid nanoparticles. So I thought I'd take some time to explain what that is. So CRISPR uses the sequence of A's, T's, and C's and G's to make what's called a guide RNA, which can interact with the target gene that needs to be changed. You then put that together with what's called a base editor, which will chemically change that base, that A, T, C, or G, so that it looks like the one it's supposed to be. So this is a personalized editing of the mutation. It's absolutely critical to have the gene mutation information there. And you also need to minimize off-target effects. So the guide RNAs tell that base editor where it is it needs to target in the genome. And at the moment, the way of getting those to work is basically trial and error, that scientists need to design a bunch of these guide RNAs and see which one is going to go and fix that mistake without introducing a load of other mistakes elsewhere. So that's why it's important that the gene mutation information is available so that these guide RNAs can be designed in advance for the mutations that end up causing Lowe syndrome. Okay, so that's the CRISPR part of this. Now I'm going to explain the lipid nanoparticles part of it. So that came out of the COVID vaccine technology. These are ways of very efficiently bringing genes or other big things into cells so that you can express this base editor. And at the moment, that targeting is most efficient for the liver, which is the genetic disease that was altered, fixed in mid-May. So it's important for gene therapy that the right tissue is targeted in sufficient quantities that it makes a clinical difference to what's happening. And so at the moment, the targeting is being developed for the kidney as well as the other tissues. Now, I also call this a platform approach because the idea is, the kind of fantasy in the future will be that there'll be the genetics department and you can get your CRISPR reagent off the shelf and you can get your lipid nanoparticle reagent off the shelf. And then if a baby is diagnosed with a genetic disease, that you can mix and match those ingredients to fix that particular disease. But we're still some way off that, but at least there's the first proof of principle that we had in mid-May, and this is likely to be many more similar reports of that moving forward. So that's one way to do gene therapy. Another way it's been used is adeno-associated virus. So this is by reintroducing the correct OCRL gene. The wrong one will still be there, but you're introducing the correct sequence. And this is well investigated as a gene replacement method. Adeno-associated virus is fairly benign, yet our bodies still respond to it. So some people have been infected by the particular strains of adeno-associated virus already, and so will mount an immune response to that gene therapy. It can be that you can't do repeated dosing because there's an immune response that's raised against that adeno-associated virus that's trying to get that gene in. There can also be an immune response against the gene product itself, because if you've got your OCRL gene, it's made a misfolded protein, that misfolded protein is degraded, then when your immune system's learning what's self and non-self, then it doesn't have that OCRL gene product to learn to not recognize. So in gene therapy, sometimes the correct gene that you introduce, your body then attacks that correct protein. So there have also been challenges in the commercial viability of using adeno-associated virus as a gene therapy modality. And we know that I think Cure Lowe are hoping to sort out some of those issues, as well as the scientific ones that Youngson and Leopoldo Staiano are working on with the adeno-associated virus vectors. So one technology that Leopoldo Staiano and others are going to be using to look at this is organoid technology. So these are, you can call them mini kidneys to test if a treatment will work. And so in these, stem cells are taken and then they're put together with a sticky substance called extracellular matrix to grow kind of kidney tissue that's more complicated than just individual cells in a dish, so that it's human tissue and it has some of that same organization and structure that our kidneys do. So that we can test and develop ways to introduce new genes into into those cells, or to screen and test for other drugs that can be used. So this is what these organoids look like here, and this is a cross-section through looking at the different components of the cell within this wider tissue context. This is a very powerful technology that's come to the fore in the scientific community. Okay, so now going on to fixing the OCRL enzyme. So this is a strategy being undertaken by Claudio Aguilar. So we've got the gene here is giving rise to the OCRL enzyme that has a particular shape, a particular fold here, like the origami. If there's a genetic mutation, then in some cases, this leads to misfolded protein. And that can be helped by using a molecule called a chaperone that helps that folding process. It's like a kind of crutch to help that folding happen. There are some mutations, like the active site mutations or the truncating mutations, where these chaperones are not going to be able to help. This has to be a folding problem caused by the mutation. So this will only be a subset of mutations that can be helped by this. So chemical chaperones can cross the membrane into cells because these are small molecules, and there are some that are existing medications already. And here, having the gene mutation information, again, is critical to know which ones are going to be able to work. The other thing that comes into play with these kind of mutation-specific, these personalized types of medications, is that we're not going to be able to make animal models or lab cell models for all of the different mutations. And so that's where donating cells is going to become important. Donating cells from urine, and also it can be samples, there can be skin samples or blood samples for making stem cells so that scientists can make organoids to be able to test developing strategies on. And this is happening in Italy. Leopoldo Stano is working with patient-derived cells, and we're hoping to set it up in Cambridge in my lab, and also Claudio Aguilar is hoping to set that up in Purdue as well. So moving on to the next strategy is to correct the blocked uptake pathway to try and fix this blockage in the membrane trafficking, particularly within the proximal tubule cells, taking an approach called drug repurposing. And so I'll have a break now for the next video in the series to introduce you to the researchers in my lab that are working on the drug repurposing.
We're researchers in Jenny Gallop's laboratory at the University of Cambridge, and we ask how the structure and shape of cells maintains a balanced, healthy function. Studying these intricate processes can have wide-ranging implications, such as in disease. But we don't always know whether we'll have answers next week, next year, or the next decade. On a day-to-day basis, we're craftspeople, data wranglers, and storytellers. We grow and manipulate cells in the lab, and take images and movies of them to find out how they are responding. We scrutinise our data and ask questions to try to find patterns in how the cells behave. We're constantly learning, discussing back and forth, and teaching each other. We share the stories of what we find to other researchers and the wider community. We navigate through the grey, grappling with things on a logical level that may not make sense at first, ultimately teasing out the truth. It can get easy to focus on small details. Working on Lowe syndrome makes us look at the big picture, that research, fundamentally, is for helping people. We hope that understanding the underlying molecular mechanisms is going to generate treatments. We're striving to get better at communicating our research and what it is saying, and in turn, to learn from patients and families about which aspects of our research will help them the most.
Okay.
We could do that.
So we've been working on a medication called alpelisib, and this affects the special types of fats. Now, piclidenoson is the drug that's been worked on by Antonella de Matisse, and that also affects these special types of fats in a different way, and both of them do it indirectly, but the sort of underlying concept is the same. So they're working at the level of the fatty substances in the membranes so that we can compensate for those changes in membrane identity. And so what we're looking at when we're studying alpelisib is this traffic jam in the cell that when you have the OCRL deficiency means that these receptors can no longer go back up to the plasma membrane surface to collect the protein for the uptake. And so the drug repurposing efforts, what we're doing is looking to resume that membrane traffic within those cells so that it restores the function of the proximal tubule. So the reason why we take an approach of drug repurposing is because drug discovery is long and expensive. Can anyone guess for how expensive this is? Any guesses here? You think a couple of million, okay. Hundreds of millions, yeah. So a couple of million will get us to kind of this bit here, which is kind of figuring out how to measure it and beginning to test the molecule. So working out what to do and trying to get a few candidates. So what has to happen then is working out which ones might work in people. That, is it toxic? We need to do there's lots of animal tests that are needed. Which ones are you be confident to give to a person? And then it needs to be tested in healthy volunteers. That's a phase 1 clinical study. So at that point, we're at 30 million. Yeah. Okay, and then it's a phase 2 trial. So that's starting to look at efficacy. So is it actually going to work? And then if you get an indication in phase 2 that there's something to work on there, then it goes into phase 3 with large numbers to see if it will work. And then you have the regulatory approval steps and then post-approval review. So many of these drugs actually don't make it through this whole pathway, and so then you get up near a 1 billion for successfully developing a drug. So what drug repurposing does is short-circuit that process. So if we take a drug that's already been developed, then much of this has already been done. If the target can be identified that's a valid target in a different disease, that's a good thing because you can reuse the same medication. And so what we've been able to do is jump into phase 2. So drug repurposing uses a drug that's already approved for another disease, which researchers are showing can correct the consequences of lacking the functional OCRL enzymes. And this can be in cells and also in the mouse models to look at the actual physiological change. Claudio Aguilar has come up with a combination of rapamycin and rovastatin, and the next step with this is looking in animals and in organoid models to see if it works. Piclidenoson from Antonella de Matisse has already been looked at in animal models and in organoids. Piclidenoson is an anti-inflammatory drug that's currently in development for psoriasis, so it's not yet been approved. So it's in phase 3 trials at the moment for psoriasis. Alpelisib, which is the one that my lab's been working on together with Olivier Devois' lab in Zurich and Francesco Emma in Rome in Italy, is an approved medication. It's already been used at long-term low dose in children in another rare disease called PROS, and it was originally developed as a cancer drug. So it's available in different doses. Here we go. So piclidenoson, I mentioned it's in development for psoriasis. They did a press release that they're seeking ethical approval for a phase 2 study in Lowe syndrome because they're in the final stages of their phase 3 trial. And this is being taken forward by Can Pharmaceutical Company together with Antonella de Matisse and also Francesco Emma, who we're working with as well. So alpelisib is an approved medication. It's 1 tablet that can also be crushed in a drink per day. It's available in both low doses and high doses, and it will be the low dose like the disease PROS. And they managed for PROS to do a real-world trial. This is comparing the chart reviews. This is similar to the information that Bob Nussbaum was saying, that comparing with the past record, comparing what the drug does when it's, rather than having a placebo-controlled trial, you compare before and afterwards. So this is an innovative trial design. It takes persuasion for the regulators to accept a study like that, but it has been done for repurposing alpelisib for PROS. So these studies had 50 patients showing that there was a positive effect in efficacy, and it also had an acceptable safety profile, as well as in children. So to pursue our work with alpelisib, we considered with Francesco Emma and Olivier Devoist, and chose to start with DENT2 adults, and that's because we understand what this drug is doing in the kidney, but don't yet understand what it might do in the other tissues. So DENT2 disease is also caused by mutations in the OCRL1 gene. These are different mutations. It ends up being a sort of milder form. The kidney disease is very similar, but it doesn't have the multi-system involvement to the same degree as Lowe syndrome. There are some intellectual disabilities, for example, some growth effects, but largely affects the kidney. And so this is a kind of a sort of safer way of trying what was originally a cancer drug within a population that has OCRL1 mutations. So this is to mitigate some of the potential risks. And that's a theme throughout clinical trials, that you need to do this on the least vulnerable patient population that is expected to show an effect. So I'm now moving on to part 3 of the talk, which is talking about this trial that's looking to improve the abnormal kidney function in DENT disease 2. It's called the ALPA-DENT trial. And this is the first clinical study that's been done in patients with OCRL1 mutations. It's a phase 2 study looking for both safety and efficacy, and it requires a specialist clinical trial centre for this to run. And in looking around Europe and thinking about how we could do this trial, was basically one place we could do it with one clinician, and that's Francesco Emma, that has that motivation and ability and the clinical trial centre to run this trial. He's got a colleague of his helping as well, Barbara Ruggiero, and Marina Vivarelli is the head of the clinical trial centre, and they've been working together to get this trial off the ground, taking place in Bambino Gesù Hospital in Italy. So what you can do here is look out for, if do you know whether you're in a specialist clinical trial centre, and whether your clinician could run such a trial in future, and if they have that potential capability, co-opt them to the cause for this, because this is a rare type of clinician that can actually do this type of study. So the identification of alpelisib built on the long history of research that was funded by the LSA. So I directly built on the observations of Antonella de Matisse back in 2005 and 2013. She was funded by the LSA. It absolutely required the mouse model that was made by Robert Nussbaum, who was funded in 2013 and 2015 by the LSA. I also used the work of Pietro De Camilli to come up with the idea of using alpelisib. And now, thank you very much, I'm being supported by the LSA as well, and so is Francesco Emma to run the trial. And importantly, the other people that were also being supported by the LSA over the years, is I've been in this field for 20, 25 years now, and I've read these people's work on a lot of occasions. So you've made strategic investments that have really given rise to a good outcome here. And of course, this is also underpinned by the bigger grant funding bodies, such as the NIH, Wellcome Trust in the UK, who funds my lab, Medical Research Council, and the EU and European Research Council funding has also been important in this. Okay, so how to reach clinical studies for rare diseases. So one challenge that we dealt with over the past 5 years since the initial paper was published is in funding. So clinical studies are very expensive, as we saw from drug development. So to run a phase 2 trial for 10 patients in multi-site between Europe and the USA would have been about $2 million. And so that wasn't really feasible or possible. So what we did was do a pilot small-scale phase 2 study at a single site to try and get the initial data an initial indications of whether it's going to work, to then leverage that information to get more money later if it's successful. So we design the study so that we, get the first idea, may it work, or because if it's not going to work, then it's a difficult thing to find this amount of money. So here, what you can do is fundraise to support the pilot studies, with the view of increasing the likelihood that promising candidates will be considered for future larger funding for the wider study. The next challenge is difficulty in designing an appropriate study, because you need to have some measure to determine whether or not it's worked. And here we need more information about the disease natural history, where giving your information to Citizen Health will help those efforts. So we need to have valid outcome measures that can be assessed of whether any drug that's tried is actually helping things. So here we've got kidney scans have been approved by the regulators as a measure for telling whether alpelisib is going to be effective or not. So the next challenge in reaching clinical studies for rare diseases is that usually young children respond much better, but many drugs are only tested on adults. The pharmaceutical company doesn't generally test. A lot of drugs in pediatrics are being used off-label based on what they do in adults. What Francesco Emma has figured out by working with his patients are that young adults here are a medically viable first approach for the proximal tubular dysfunction, that the GFR is okay to be able to see an improvement. So this is good news that the adults were potential participants in these studies as well. So here the action that you can take is carry on working hard. I know it's hard from hearing what all of you are saying, to preserve that kidney function with your nephrologist, because there may well be options that become available in the next 5 years. So for the participants in the Alpident trial, they need to have a genetically proven diagnosis of Dent disease 2. They need to have the adjusted GFR of more than 50. So I think this is a stage 2 kidney disease. They need to have no serious medical issue other than the OCRL mutation. In particular, they can't have diabetes, because what alpelisib doing is what's called a PI3 kinase inhibitor, and this used to an elevation in the blood glucose level, so you can't make that diabetes any worse. But usually when people are on alpelisib, that this can be managed by diet. So people need to also not have a hypersensitivity to any of the ingredients of the alpelisib pills or the radioactive tracer that's used for measuring kidney absorption. So the endpoints, the outcome measures that are being used in the Alpident trial is primarily the kidney scan. That's the first one. This is what's called a DMSA scan, where there's injection of a radioactive tracer into the blood that's then absorbed by the kidney. And we're looking for that increase in absorption after the patients have been on alpelisib for 1 month. And also the kind of blood tests and urine tests that Eileen Brewer described yesterday, to see if they're going in the right direction over this short period of treatment. So what you can be doing is thinking about any adaptations and measures that you would want to be endpoints in Lowe syndrome, because it's the more complicated multi-system involvement. So think of, co-develop and consider, what kind of outcome measures you would want, whether this is caregiver impacts, behaviour scores, or other things. So we're running the Alpident trial according to good clinical practice. These are a set of rules that are used internationally for testing medicines. And these, it involves people have to be honest, they have to get really genuinely informed consent, it needs to have regulatory observation, the science needs to be transparent, there needs to be advice taken on how to run this study, and the appropriate data privacy and archiving, so that information is recoverable later and can be used later. So it's more expensive to do things according to good clinical practice, but it helps make sure that this is done safely and that the data is valid to put forward to regulators at the next step of getting approval. So in order to make all this happen, we've been talking a lot. There's been a lot of discussions and connections over the past 5 years with the LSA, between myself and the science lab, Olivier Devoist in his lab, who did the mouse model, and the clinicians. So the last video is about how this connection can circle around and focus on the patient group that has a key role in bringing us all in contact and reaching this cure that we all want. So this is now the last video in the series.
Coretta Scott King once said, "The greatness of a community is most accurately measured by the compassionate action of its members." This gets to the heart of the Lowe Syndrome Association's mission. We're a community of people who have come together to take compassionate actions to help each other achieve some very specific goals. We want to build champions and awareness. We know that we can't make progress in helping families and funding life-transforming research if no one even knows about Lowe syndrome. That means we need to raise targeted global and local awareness about Lowe syndrome where it matters most. It also means we need to identify champions who can be advocates for us in governments, schools, the scientific, and local communities. Second, we need to support our families with meaningful resources. This means providing valuable and practical information, tools, and connections to families and circles of care from diagnosis and every significant milestone throughout life and beyond. And finally, we want to achieve our goals of research funding and advancement. We know science holds the answers we need to make our boys and young men healthy. We are optimistic and ambitious in our strategy to uniting the scientific and medical communities to find real-world treatments and a cure for Lowe syndrome. But to succeed, we need to do 3 key things. First, we need to be aligned on these shared goals around awareness, support, and funding research. Second, we need to work together to move smartly and quickly to empower progress. And finally, we need to collaborate, learn, and then share what we're learning with each other. Then repeat. This is how we'll make progress. This is how we'll achieve our mission. Margaret Mead once said, "Never doubt that a small group of thoughtful, committed citizens can change the world. Indeed, it's the only thing that ever has." We invite you to join the Lowe Syndrome Association and help us take compassionate actions that will change the world for the better.
So we've had 5 groups talking with each other and getting together to make sure this trial got done. It was the Dent Disease Foundation, the Lowe Syndrome Association, the Lowe Syndrome Trust based out of the UK, Bambino Gesù Hospital in Rome, with Francesco Emma, as well as myself in Cambridge, Olivier Devoist in Zurich. So what can you all do? So I'm now recapping the points that I've said through the talk, and that's to find good clinicians. If you're in one of these specialist trial centres, see if your clinicians would be able in future to run such a trial and co-opt them to the cause of finding people that can actually do this work. You can get the sequence information and register with Citizen Health to give that natural history information and the precise mutation information of how your child's affected. When the projects are up and running, hopefully there'll be the opportunity to donate cells so we can look at these cell biological changes directly and make the approaches that will fix the particular mutations. And then communicate what you think would be the meaningful measures beyond the kidney that you'd want to see in future trials. So thank you all very much for your attention. I'd also like to thank everybody that's helped me put this talk together. So the professional services staff in the Institute, so Natalie Wars worked together with us on the video project and also helped make the handout. So I think Teresa's come round with some of the leaflets because the feedback that I got from my institute was absolutely you have to give them something, some handout, so that you can recap that information, they can access it and get back to it later. So I hope that's helpful. together with Helene Dorflinger, who's also in our public engagement team, and the people in my lab that are doing this work. Bambino Gesù Hospital, who are running the trial, and Francesco Emma, who's been an absolutely wonderful collaborator. The families that have worked together with us, Teresa Huygen in particular, also Paul James and Wendy James in the UK, Morag Donald, and Jerry Kubicki, who puts up with a lot of my emails. On the Lowe research side, I requested information and permission to talk about their work from Claudio Aguilar, Youngson, Leopoldo Stano. Robert Nussbaum gave me feedback on the talk, and also Olivier Devoist, a long-term collaborator. Thank you very much.